Laura Chirica, and I am CEO of Cellevate since 2022. I was assigned by Cellevate Board of Directors in 2021 as a strategy consultant to evaluate the use of Cellevate core nanotechnology in industrial biomanufacturing of novel therapies, such as gene therapy, novel vaccines, cell therapies. I come from a scientific background with a long time ago, PhD in biochemistry, but with 25 years experience in the business and commercial growth by creating and implementing successful commercial strategies for large corporations in the exact same bioprocessing space as Cellevate, such as Cytiva, that was called GE Healthcare at the time, Sartorius, Agilent Technologies, which was Deco at the time, as well as medium companies with laboratory biopharma services, which is now called Svar Sciences, and startups in cancer diagnostics with Immunovia and also now Cellevate. Yes, that is actually a very good and relevant questions, especially talking about vaccine manufacturing. So, if you think about in general this question 2D versus 3D cell culture in vaccine manufacturing, the vaccine industry is and I'm coming back from World Vaccine Congress last week, so they are still largely relying on 2D systems such as tea flasks, even roller bottles, cell factories, cell sacks. But 3D cultures are emerging as a much more efficient and scalable alternative. Talking about 3D cell cultures, now using microcarriers, in our case, nanofiber-based microcarriers, enable much higher cell densities with high cell viability, much higher virus titers or yields, and support high scalability production processes in stir-tank bioreactors, either way, that are single-use or stainless steel. They provide the so-called spheroid growth structures that are formed by the cells and the nanofibers, having this very high cell density and viability. This spheroid culture improves the reproducibility of the process and the scalability, and they are compatible with the growth in from the benchtop bioreactors to the very large scale ones, and then provide automation. However, you asked about limitations, and I think you know what I experience, and now we have been pre-launching and working with this for quite some time, and coming from working in this field for so long, the introducing to biomanufacturing, such a long time kind of established production. 3D systems that we are now coming up with, they introduce a completely new way of cell culture upstream bioprocessing. And, this is always the curse of being first and innovative. You will require process optimization, right? And you will require to change. And then we need to convince the process developers, the production teams, that this implementation of the nanofiber-based cell culture innovative way is faster, is easier, is much more productive, is having higher sustainability, being based on cellulose, low waste, low water use, and so on. On the other hand, additionally to that, there is the regulatory. And the regulatory familiarity with the 3D systems is, in this case, developing. And all of this can and will probably slow adoption a little bit, especially in the beginning. Now, I think as technologies like Cellevate 3D VAX, nanofiber microcarriers mature, the 3D systems are rapidly becoming a viable and superior alternative for this industrial scale production of vaccines, especially viral vaccines and viral vector-based, because they offer the benefits that are suitable for the next generation, what they call it, the second generation vaccines and viral vector production. And you always look for higher productivity, scalability, And in the end, these are all connected to the regulatory alignment with whatever this industry needs for the future. And actually, the future is here. It's also a good question and reflection now that we enter with a completely new way with our nanofiber based products. Um, there are several key challenges, and there is actually, and we could see it all this time when we have been in contact with different types of vaccine producers and CDMOs, and there is a strong legacy dependency on the 2D systems. There are facilities, there are processes, there are regulatory filings based on the flat surface cultures, making them at least I could see in the beginning hesitant to change. Then process optimization, as I said, is very important. And in this case, it includes everything from new protocols as well as new analytical methods that characterize growth, product yield, and then thirdly, there is a limited standardization in terms of the also the regulatory, and that will slow down the validation and the approval processes. But I think what we have been very much working with several of these partners is so important to work in partnership to bring this kind of solutions to the market is to overcome them. Then you do look at bringing together solutions. So you take the Cellevate 3D platform products, the VAX product, for example, and you put it together with ready-to-use bioreactor formats, and then you work together into a solution with improved process economy. And then in this way, you enable an easier integration and acceptance from the very beginning into the existing GMP workflows, and also showing that in this way you are going to reduce the gap between the 2D and 3D, especially in their current processes. I think, as always, and this is very common when working in biomanufacturing, especially bioprocessing that we are now providers to, comparative data, benchmarking and comparative productivity, consistency data. So as more of this become available, and then their comfort is increasing by seeing in their evaluation processes the fantastic results, then 3D cultures with products innovative like Cellevate 3D Vax is likely to become a standard for this high density, scalable, high productivity vaccine production, particularly for viral vector-based vaccines. Also, immunopluripotent stem cells therapy and other types of advanced biologics, recombinant proteins, organoid production that fit with this kind of nanofiber-based platform. Every year, CPHI is such an important event for all kinds of reasons. I really appreciate the organization of CPHI, and this year is not going to be different in any way in Frankfurt. I'm most looking forward to connecting with partners across vaccines and any kind of biomanufacturing novel advanced therapies ecosystem, from CDMOs, media suppliers, technology providers, bioprocessing distributors, established distributors. We are in a commercial stage, and we are very happy to be in that stage. And it's an ideal opportunity for us to discuss how advanced 3D cell culture technologies and products like ours can help address the industry's growing need for scalability, for high yield production systems that have a significant impact on their cost of production, because we think about vaccines here, and we think about the possibility to produce and give access to as many patients as possible. So it becomes an extremely important point looking at discussing the needs of the different partners and addressing them with our products. I'm also very eager to explore, because when meeting with all these people, and it's usually so easy to get in contact as they are all present there, to explore the latest developments in upstream bioprocessing, in vaccine manufacturing, in next generation advanced therapies, to exchange. Usually we discuss about, how did you get your automation in place, how is your sustainability, how are the new production technologies that you are using, how do you see that this field is actually growing and changing and developing towards what we call the biomanufacturing revolution. Are we getting there, or are we just getting started? What is it missing to get there? And I think, for us, it's so important that CPHI brings together the people who are making innovation happen. So I'm looking forward to building this new collaboration and accelerating the kind of the real world adoption of the 3D bioprocessing solutions as Cellevate 3D nanotechnology.